All right, adaptive immunity. So again, this is specifically, you can't sit with us. So creating a defense against a specific one. So with our immune system, you're building a defense against a pathogen once you've been exposed to one. And with adaptive immunity specifically, it is more systemic. So with your innate immunity, you're going specifically to that area to target that pathogen. While with adaptive immunity, part of your defense is going to create clones or antibodies that will circulate throughout the entire body. It's specific to a particular pathogen, and unlike with your innate immunity, you can actually remember an infection and have that defense all ready to go if you get exposed to that pathogen. So there's two types of adaptive immunity. We have cellular and humoral. So cellular is cell-based. Humoral is fluid, humors-based. So with cellular mediated, the lymphocytes are going to actually directly attack the foreign pathogen. It gets rid of it in areas where you don't have antibodies able to get to them. So, for example, inside a cell, so like the natural killer cells directly attacking that foreign pathogen, or that cell's been affected, you're going to where the antibody just can't like budge its way through a plasma membrane to get inside a cell. With your cell-mediated immunity, you also can kill the cells that harbor any type of foreign pathogen. The humoral one, so body fluids, that's just an old way of saying humors. Humors is an old way of saying body fluids. So the antibodies are hanging out in those body fluids. So this is a extracellular stage where the pathogen hasn't found a host, and so those antibodies can bind to that pathogen and therefore start the process of the immune response. So both cellular immunity and humoral immunity can be found in adaptive immunity. In both of them, the immunity occurs in 3 stages: recognition, attack, and memory. Also thought of as the 3 R's of immunity: recognize, react, and remember. So for cellular immunity, doing adaptive immunity, so the specific defense, you have your T lymphocytes. Those are going to work as an antigen presenting cell. Once they've attacked, they can also remember your foreign pathogen in the future. So again, you already have a defense against something you've been exposed to. And we already talked about these earlier: your cytotoxic cell, your helper, and your regulatory, and your memory, and what they do. So one thing that they're able to help with in our 3 R's of immunity is to help to recognize a foreign pathogen. So our T cells can work as an antigen presenting cell. So process that foreign antigen, display that foreign antigen on its surface, go to the nearest lymph node so it can activate some B cells. So their job is to initiate your immune response. So start everything getting to going. So again, we already talked about this earlier. Those major histocompatibility proteins. So that's where it displays the foreign antigen. So when a T cell finds a foreign antigen which is also on the major histocompatibility complex on that foreign antigen, that T cell can read that, recognize it as foreign, and then essentially break off that flag and display it on itself so that it can alert the immune system. So the type of protein, don't really know this, but it's the MHC2 one that it displays for foreign antigens. Okay, our last 2 for our cellular immunity is react. So our cytotoxic cells for T cells, those are the ones that are going to directly attack and kill the pathogen. And then lastly, remember, our memory cells, those are the ones, again, you already have your defense clone of army against that specific pathogen, so it remembers the next time you're exposed to that pathogen. The other type of adaptive immunity is humoral immunity. So this is with those B cells or B lymphocytes. So their job is going to produce the antibodies. The antibodies are going in your body, humor, your fluid, and it has those 3 stages: recognition, attack, and remember for your 3 R's. Okay, so first one, gotta recognize. So with your immunocompetent B cells, so these are the ones that can detect the foreign antigen on a antigen presenting cell, like a T cell. So once it binds to these foreign antigens, so it binds to several of them, it can also display them on their own cell surface once they've been activated. But they need that T helper cell in order to get them to going. So there's our tag team again, which helps to, that T cell is going to secrete those interleukins so that that B cell can now actually go through the whole process of making a bunch of clones through mitosis and either becoming a plasma cell or a memory cell. So there's our mitosis, our B cells that are genetically identical to the original one that got activated by that T cell. All of those are, again, they will respond specifically to that foreign pathogen. Some become your plasma cells. So all these little pink squiggly lines that are off to the corner, so showing the extra endoplasmic reticulum. So those are going to make proteins, and there we go, good. Make those proteins, which are the antibodies for killing off an infection. There we go. And then you do have some that become your memory cells, so just in case you get exposed again, you got your defense up. So the attack portion in our humoral immunity is the antibodies that are made from the plasma cells. So here they're showing all that excess endoplasmic reticulum. So with all the ribosomes attached to them, so the rough endoplasmic reticulum to make all those antibody proteins. So when those antibodies bind to that foreign antigen, they're going to tag it, and that's going to activate other immune cells to destroy that foreign pathogen. And then some are just going to be memory cells for next time. For hopefully not next time, but anyway. So there's 4 mechanisms of attack for humoral immunity. So we have neutralization, complement fixation, agglutination, and precipitation. We've already covered agglutination from our last section, by the way. Okay, neutralization. So the antibodies will bind to what's called the pathogenic region of an antigen. So basically the part, for example, let's say if you had an antibody against the COVID virus, it would bind to that spike protein that is needed for that virus to enter the cell. So basically you've kind of put it in handcuffs or made it not able to cause a disease response in your body. Next one is complement fixation. So this is where those complement proteins bind to that antigen and then increase or recruit other complement proteins in there, leading to what we talked about earlier: inflammation, phagocytosis, immune clearance, or cytolysis. So this is how our body focuses against anything that's like foreign cell, bacteria, or those mismatched red blood cells. So that antibody binding to it. So particularly that one, your agglutination, you have your antibody, they're binding to the foreign blood cells, makes them all sticky, keeps them from spreading around. It also helps with phagocytosis because it makes a nice big clumpy bite of red blood cells that can be phagocytized or eaten up. And then the last one is precipitation. So the antibody binds to the antigen, so the foreign cells. Then that foreign cell, again, using immune clearance, so hitching a ride on red blood cell, or because that antibody has basically targeted that foreign cell, then they could be phagocytized by an eosinophil. Okay, so with our antibody creation, the first time we're exposed, so that's our primary immune response, you only get so much of a antibody buildup to the first time, partially because some of your antibodies are actually being used to fight off the infection. Plus, you don't have as many initial activated cells because you just have that 1 or 2 or however many you had that got activated. So they haven't built up that clone army. So not too many antibodies are made. It's also kind of a slow process the first time, but the second time, whole lot faster because you have a whole army of cells that were cloned during that initial infection or initial vaccination, whatever. So all those clones can create a bigger response and a whole lot faster response. So here, even the pink peak of the blue, first time it's like after 10 days, but the second time it's between 5 and 10 days. So faster response, especially the second one with the immunoglobulin G. So I didn't put this in the PowerPoint just because y'all have so much to learn already on this chapter. But you have 5 different types of antibodies and they have letters. So immunoglobulin M is one of them. You also have an A, D, G, and E, and they all have their specific jobs, but y'all had enough to deal with this chapter, so I didn't put that in there. But one way to remember that is MADGE, M-A-D-G-E. Okay, so second time, faster response and a whole lot greater response. So because you have a faster and greater response, typically no illness happens. You're technically immune to it, even though you are infected the second time, or you just have a less, a smaller infection response to that second time. So less symptoms the second time around. Okay, so with our immunity, we do build it up, and there's different forms of them. So it's either an active or a passive form, and your defense is produced either naturally or artificially. So this ends up with 4 different types: natural active, natural passive, artificial active, artificial passive. So we have our 4 different types here. So let's first look at active versus passive immunity, and then natural versus artificial immunity. So active immunity was your body produces the antibodies on its own. So you've been exposed to the pathogen, and so your body has to work and do its own production of the antibodies. Passive immunity is when the antibodies are produced in someone else's body, and you just receive passively the antibody. So one of the treatments for COVID, if you can get it, is a collection of antibodies that were produced by people who survived COVID. So that's a passive immunity against that COVID disorder. Then with your natural versus artificial immunity. So natural immunity is where you just happen to get sick. So you actually just get exposed to that pathogen, versus artificial is, for example, when you are exposed intentionally, or it's like for example, through a vaccination where it's a part of the pathogen, not the whole thing, where your body can make up the defense so you're ready to go for the next time. So, examples of this: a natural active immunity. You have been exposed to that pathogen by chance. It just happens, so you naturally get infected, and you are actively making your own defense against that pathogen. Artificial active immunity is where you intentionally expose yourself so you can build up a defense, and it is active, so your body is making that defense. So this is why we get those vaccines, so we can control the level of exposure. So again, you don't get the whole virus, for example, for COVID, but you just get a piece of it, enough to trigger the immune response, but not the whole thing that actually causes an infection. So when you stimulate the immune response, you don't cause the disease, you're just building the defense against it. For some vaccines, you do have to get booster shots to stimulate the immune response again, to kind of, hey, you've had some soldiers die off, so let's make sure you get some more of those clones going in your body again. So for example, the tetanus shot, you're supposed to get that every 10 years because those cells will, of course, die off some. So kind of peak up your production of those memory cells so that you're ready just in case you are exposed. This will also happen if, like, you have, like, you step on a, well, it's not the rusty nail, it's the dirt on the rusty nail. That could be like the tetanus bacteria. And so, you know, you go to the hospital and they give you a tetanus shot so that you're like, hey, let's stimulate the production of those memory cells, those antibodies against it, so you have a better shot at not having the disease from the tetanus bacteria, which is actually deadly, just because your body spasms up. So remember the tetanus from the muscle chapter, the tetanus happens while your body is just frozen in a continuous muscle spasm, which includes your diaphragm, which means you can't breathe. Okay. Next to natural passive. So this is where the antibodies are produced by another person. So for example, let's say and this is kind of really cool when it deals with breastfeeding. So like if the mother gets sick and she has, her body's producing antibodies, some of those antibodies will go into the milk, and so therefore it gives the baby a protection against whatever the mother's sick with. Another thing is if the baby gets sick somehow during that whole feeding process, or maybe it's just because of that saliva exchange from the baby into the mother starts making antibodies to eventually pass into the breast milk, and therefore passing those antibodies back onto the baby. So the baby, again, your defense system really hasn't started yet as you're that young. So this helps them to fight off that disease. So there's literally, you can just Google this. Look at breast milk, like with an infection and without. And without, it's that kind of normal white-ish color. And I saw one lady's, she had gotten COVID, and her breast milk was actually green just because of all the antibodies in it. Anyway, next one is artificial passive. So the antibodies are made in another person or animal. And this is where you're just given a treatment, kind of like with, for example, like with COVID, where, where the antibodies are just given to you. So we have some against different types of emergencies, such as a snake bite. So you have antibodies against the venom in a snake bite. Botulism, which is a bacterial toxin. Same thing with rabies, tetanus, and other things where we just give you antibodies for a treatment for whatever you just ended up with. Okay, going over some definitions just for some of our terms. So a antigen is anything that can trigger an immune response. So this, again, this is unique to the individual. So even on, for example, the antigens on red blood cells, they can still elicit an immune response, so that agglutination. So helping to form that glycocalyx, again, unique to the specific person, unless you have an identical twin. And this allows our body to determine the difference between something that is a self molecule versus a foreign one. The epitope is the specific region that causes that actual immune response. So that specific area that is different, that's going to stimulate that response. Now, some stuff, it's going to have some kind of an immune response, but it's not actually being like, you don't actually have a disease, but you still get a response to it. So these are called haptens. They're too small to actually elicit a whole immune response, like a disease, but you still, so a hapten's too small to actually cause a disease, but it can trigger an immune response. Remember our weird kids, they fight off allergies? Same thing. So it's something foreign, the body recognizes it as something foreign. So it could be like some like cosmetics, so like makeup or something some people are allergic to, different detergents, chemicals, different plant compounds like poison ivy, animal dander, pollen, dust. Those things can trigger an immune response in the person, so they have an allergy response to them. You can give them penicillin, like for some stuff, or you have the antihistamines, or like for example, your steroid asthma medications that help to kind of fight off your body from doing the immune response to whatever your allergen is. Okay, so looking at your antibodies just generally in the structure, so some of that stuff is the same, but then it's these outer binding portions that are what's different between your between the antibodies that are specifically binding to your foreign pathogen. Okay, so this last bit on antibodies, I'm just putting it in there just in case you need it for later. At least you're exposed to it, but it's not in the test. So this is what I was talking about earlier, MAJ. So you have different types of antibodies. And again, the difference between them is the part where it actually binds and does its job. But it's that region that is different, that makes that causes different little classes. So each of these have their own different type of response of what they actually cause for your immune system. So that's why they're in their different categories. But again, I'm not going to cover them because they're not on my test. That's less time necessary. So again, it's all just genetic just random creation of getting different regions that are shaped differently to bind to different pathogens. And that is it for this chapter. So finish up that study guide, and again, ask me questions if you need to. That's kind of what I'm here for. Duh. So I'll see you guys in the next video.